Yes, we know that when we use the COMET assay in cells which derives from human subjects, we can approach different kind of studies, such as human biomonitoring studies, molecular epidemiology studies. As in these studies, as already stated by Stefano before, the COMET assay is a tool that is useful to identify the effect of the extent of exposure to genotoxic xenobiotics and the effect, the genotoxic effect as a consequence of the exposure. And these tools are, as the name of biological markers or biomarkers. And the COMET assay is recognized as to be a useful biomarker in this field by virtue of its role in carcinogenesis, as a primary DNA damage is an initiating step. This is a scheme that is based on that presented before by Stefano. And within this context, the primary DNA damage as evaluated by the COMET assay represent a biomarker of biologically effective dose, thus indicating the extent of interaction between genotoxins and DNA. And on the basis of the study design, the authors consider this biomarker more associated to the exposure to better define the exposure level, or sometimes as a biomarker of biological effect. Also, in this case, also Stefano has already stated that the best scenario is that the biomarker is evaluated in the target cells or target issue. But sometimes this is not possible, so we have to use surrogate cells. And surrogate cells, the most used surrogate cells are peripheral blood lymphocytes, leukocytes, because they are very easy to obtain. Blood withdrawal from the antecubital vein is very easy, and it is a procedure that is common in health surveillance in population with occupational exposure, not only to genotoxic xenobiotics. However, even if this procedure is not very invasive, there are also situations in which blood withdrawal could not be accepted by population because of the absence of risk perception. As in the case of occupational exposure, the subject may not have a known exposure to genotoxic xenobiotics, or in the case of residential exposure, because the people is supposed to not be exposed to pollutants, and they may consider this approach too invasive. The same, peripheral blood withdrawal could be difficult in children, could be not accepted by children themselves, and also it could be not authorized by the ethics committee. So we have to move toward different surrogate cells, and buccal cells are becoming increasingly popular in human biomonitoring studies because they can be obtained really non-invasively and can be used in several approaches to assess DNA damage. For instance, buccal cells are probably now the more used cells in the micronucleus, in the cytome micronucleus assay. But buccal samples contain mixed population of cells, epithelial cells, fibroblasts, erythrocytes, leukocytes. And so we have different yields and proportion on cells depending on the isolation procedure and the physiological state of donors. So in this approach, in the MAPEX study, monitoring air pollution effects on children, we have tried to limit our interest to buccal leukocytes. And we have tried to obtain buccal leukocyte by mouthwashes. Children were asked to wash their mouth with the normal saline, and we collected the mouthwashes in conical centrifuge tube. Leukocytes were centrifuged, were washed, and then resuspended in agarose. But we had some problems because these are examples of our slides. Together with the leukocytes, we had a lot of buccal mucosal cells, and we had a lot of debris, main food. So we have addressed our attention to salivary lymphocytes. And there are very few studies reporting this approach in the literature. And based on these studies, we have proposed our approach. The first step is about the same as first. We have collected in voluntary subject, a student, university student in this case, we have collected the mouthwashes. But in this case, the pellets was not used to prepare the slides for the COMET essay, but the pellet was subjected to sucrose density gradient centrifugation to obtain mononuclear cells, mainly lymphocytes. On the same subject, we have also performed blood samples withdrawal, because in the same samples we have performed the COMET assay in buccal lymphocytes, but also in peripheral blood lymphocytes, isolated with the density gradient centrifugation, the same. And we have also performed the COMET assay on wall leukocytes by resuspending in agarose an aliquot of wall blood. And the same, we have collected capillary blood samples, and the blood was used to analyze in the COMET assay wall leukocytes. The main results, it is, of course, the number of cells we have obtained is very different. However, even very low, we have obtained a suitable number of cells to perform the COMET assay with the buccal salivary lymphocytes. You can also see that if the viability of the peripheral blood lymphocytes is always very high, viability of the buccal lymphocytes is lowest. And the same, we have obtained very high value of basal DNA damage in salivary lymphocytes compared to peripheral blood lymphocytes. Anyway, in the Blend-Altman analysis, results indicate that the two procedure provides similar measures as the level of agreement lies within the accepted range. So even though there is a difference in the extent of basal DNA damage, the information is about the same in the subject. And so the use of salivary lymphocyte in exposure assessment is suitable. As expected, buccal lymphocytes showed a lower viability and higher levels of primary DNA damage when compared with the peripheral blood lymphocytes. We suppose that this is a consequence of the stressors, the stress to which are exposed the buccal lymphocytes, as they are exposed, for example, to air, to redox stress of oxygen, and so on. And furthermore, the higher aspecific damage that we have observed in the COMET assay may be due to the procedures in which saliva samples are exposed to environmental stress, for example, mouthwash, mouthwash for a longer time compared with peripheral blood lymphocytes isolation. Anyway, the sampling method is totally non-invasive, quick, cheap, and suitable, and well accepted for all ages. And so we think that, of course, the procedure needs to be further validated in a wider population. Reproducibility and performance of the procedure should be assessed, for example, in multicentric studies. We have to take into consideration the effects of confounding factors, smoking, diet. Even considering all the mentioned limits, we actually believe that this procedure could represent a useful and reliable tool for future studies in humans. Thank you very much for your attention.
I tried it myself with the saliva and I always get a lot of background damage. How did you manage to get such nice round comets without background damage?
Yes, we think that the main problem in this procedure is to find an equilibrium between obtaining a suitable number of cells without stressing the procedure, for example, centrifugation, duration, speed during density gradient centrifugation. Because if, of course, you may obtain a higher number of cells if you stress centrifugation, but we have seen that you induce a more higher extent of basal DNA damage. So we are trying to stress the less is possible the cells during the separation, just to obtain the suitable number.
Yeah, it was a pilot study that I did with a student, but it's very interesting because, of course, it's non-invasive and we wanted to do it with children, which is, of course, a very interesting population, as you showed already. So, yeah. So we'll have to maybe update or like make the protocol more standardized.
I think that this is a procedure that needs to be validated, of course. And so I hope that it could be possible to start with a collaboration in multicentric studies to find the better profile for this procedure.
Massimo, do you expect a relationship with oral hygiene? So people brushing teeth regularly or using mouth water or something like that?
This could be perhaps the main bias of this kind of, with this kind of cells. Because the difference in oral hygiene may influence in the subject also the stress in that environment that the cells suffer.
Yeah, it could be you can see it as a disadvantage, but maybe also as an advantage if you are interested in oral cancer, for instance.
Yes, it could be. Yes, this is an interesting approach to consider.